All right, today we're diving into something that honestly sounds like it's straight out of a sci-fi movie. Scientists have built a tiny living brain network in a lab, and then they actually taught it something. Let's break down exactly how they pulled this off. I mean, think about it for a second. Every single memory you have, every skill you've picked up, from riding a bike to speaking a language, it's all stored in the connections between your brain cells. So how does that physical network, that wiring, actually change and adapt to let you learn new things? For neuroscientists, trying to map this live, ever-changing network in a living person's brain is, well, it's like trying to map every phone call on Earth, all at the same time. It's a massive, almost impossible challenge. But, as this new research shows, they've found an incredibly clever way to build a simpler, working model of it right in a lab dish. Okay, so before we get to the big breakthrough, we really need to understand the fundamental problem they were up against, this whole mystery of the brain's long-range connections. Think of it like this. For a while now, scientists have been able to grow these tiny mini brains called organoids. They're amazing, but they're kind of like isolated towns, great for studying what's happening locally. But the brain's real power, its magic, comes from how everything is connected, like a nation linked by superhighways. To understand really complex stuff like memory or what goes wrong in diseases like Alzheimer's, you have to be able to study those highways. So the big question is, how do you build those highways in a lab? This is where the real genius of this experiment comes in. They didn't just grow one mini brain. They engineered a whole network of them, a system they call a connectoid. And this little thing right here, this is the device that makes it all happen. It's a microfluidic chip. It's basically a tiny silicone structure with these little chambers for the mini brains and microscopic channels connecting them. It acts like a tiny construction guide telling the brain cells exactly where to build their highways. The process itself is pretty straightforward, broken down into 4 steps. First, they grow the mini brains for about a month. Then they pop them into the chambers on the chip. Those little channels on the chip then guide the axons, which are like the brain's long data cables, to grow out and find each other. And over a couple of weeks, these axons actually bundle themselves together, creating a living physical network. So here's what the most basic version looks like. Just 2 organoids connected by a single one of those axon highways. The bright green you're seeing is a stain that lights up the neurons and all their connections. And even this simple link, this is a huge step forward. Okay, but what happens when you add a third one to the mix? Well, with 3 organoids, they could create a loop circuit. And this is where things start to get really fascinating. It's not just a point-to-point connection anymore. It's a structure where information could potentially circulate and create feedback loops. And this just shows you how scalable the whole system is. Here are 4 organoids, all linked up in a square. This creates a much more stable, more complex circuit that actually starts to mimic some of the looped, modular structures we see in our own brains. All right, so they've built these beautiful little structures, but the most important question is, do they actually do anything? Does adding more connections actually change the way the network behaves? The answer, it turns out, is a resounding yes. I mean, the difference in activity here is just night and day. On the left, that's the electrical burst frequency of a single isolated organoid. Now compare that to the 4 organoid network. The connected network is firing off bursts of activity way more often. It's not just 4 times the activity. The network itself creates a whole new level of dynamic energy. And it wasn't just me saying it. The researchers themselves put it perfectly. It wasn't just more activity, it was more complex activity. The patterns were richer, the bursts of energy lasted longer. The network was acting in a way that was way greater than the sum of its individual parts. So this leads to the next huge question. Is all this extra firing just chaotic, random noise, or is it actually organized? Is this network becoming more efficient, more, brain-like? To answer that, we need to talk about a really key concept called criticality. The best way to think about it is like a perfectly tuned engine. If the activity is too orderly, it's like an engine that's just idling, not really doing much. If it's too chaotic, it's like an engine revving out of control. Criticality is that perfect sweet spot right in between where the brain is running at its absolute peak efficiency for processing information. So the researchers measured this using something called the branching ratio, where an ideal brain network has a ratio of exactly 1.0. And what they found was amazing. The single organoid you can see is pretty far from that ideal state. But as you connect more of them, the network gets significantly closer to that perfect 1.0 sweet spot. The system is literally self-organizing to become a more efficient computer. Okay, so now we get to the part of the study that is just mind-blowing. Pure science fiction made real. They built a network that's more active and more efficient. But the real wow moment is this. Could they actually teach it something?
To do this, they used an incredible technique called optogenetics. It's basically a biological light switch for neurons. They genetically modified the neurons to be sensitive to light so they can activate them with a simple flash. So they took their 3-organoid loop and repeatedly stimulated it with blue light in a specific sequence. 1, then 2, then 3.
The experiment was set up just like a classic learning test. First, they recorded the network's natural, spontaneous activity, its baseline. Then came the training session, where for 12 hours straight, they flashed that 1, 2, 3 pattern. Finally, they turned off the light and just recorded the spontaneous activity again. The big question, had the training changed the network's default behavior? And they didn't just do this for a few minutes. They kept this training going for 12 solid hours. They were really trying to see if they could physically embed this pattern into the circuit's memory, to truly teach it something new. And the result was absolutely stunning. This chart shows how often that 1, 2, 3 pattern occurred on its own before the training compared to after. The difference is just huge. After being trained, the network was significantly more likely to fire in the exact sequence it had been taught, all by itself. It had remembered. It had learned. So this isn't just some cool party trick for neurons in a dish. Building a tiny learning network like this, well, it's a monumental step forward for the future of brain research. It's a huge deal. Just imagine the possibilities this opens up. What if we could build models of the specific network problems seen in diseases like Alzheimer's, or test new drugs on a functional human circuit instead of just isolated cells? What if we could finally investigate how faulty circuit loops might lead to conditions like OCD, and for the first time, actually watch the fundamental mechanisms of memory being formed right in front of our eyes? What this research really represents is a foundational leap. We've gone from just studying isolated cells to building and training functional circuits. And it leaves us with this really tantalizing final thought. If we can teach this tiny 3-node network a simple sequence, what could we someday teach a much, much bigger one?